Welcome to the Bodhi Academy. My name is Shrutika and today let us learn a new chapter from Standard 12, that is molecular basis of inheritance. So in our previous chapter we have seen what is inheritance and how the genes play a very important role in the inheritance process. We have seen Mendelian inheritance experiment wherein we have seen that certain gene brings certain characters from the parent to the offspring. So how this happens, that is how this molecular inheritance happens in human beings and in organisms. So that we will study in our today's chapter, that is molecular basis of inheritance. So basically the genes which are there carry DNA material inside it. So this DNA is very essential as it brings the genetic material from the which is that inherited from the parent to the offspring. So let us start with the chapter, that is molecular basis of inheritance. So before starting with the chapter, what we are going to study is what is DNA and what is RNA. So over the years after Mendel, the nature of the genetic material was investigated, resulting in the realization that DNA is the genetic material in majority of the organism. So what is DNA? DNA is basically deoxyribose nucleic acid and RNA is ribose nucleic acid. These are the two types of nucleic acid found in the living organism. Nucleic acids are polymers of nucleotides. DNA acts as a genetic material in most organisms, whereas RNA acts as a genetic material in viruses. So RNA mostly functions as messenger. RNA has other factors such as adapter structure as a catalytic model. So structure of the polynucleotide gene. So the nucleotide has three parts, that is nitrogenous bases, a pentose sugar and a phosphate group. So this is how the structure is. So it will have three parts, that is nitrogenous bases, pentose sugar and a phosphate group. Nitrogenous bases are purine, adenine, guanine and pyrimidines are cytosine, uracil and thymine. So there are two types of nitrogenous bases, that is purines and pyrimidines. Cytosine is common for both DNA and RNA. Thymine is present in DNA and uracil is present in RNA. So the only difference in the base pair is that thymine is present in DNA and in RNA thymine is replaced by uracil. So nitrogenous bases is linked to the pentose sugar, therefore N glycosidic linkage to form a nucleoside, that is adenosine and guanosine, etc. Where a pentose sugar is linked to the 5th OH of the nucleoside through phosphodiester linkages, a corresponding nucleoside is formed. Two nucleosides are linked through the 3' to the 5' for phosphodiester linkages to form a dinucleotide. Several nucleotides can be joined to form a polynucleotide chain, and the backbone is a polynucleotide chain is formed due to sugar and phosphate. The nitrogenous bases linked to the sugar projects out from the backbone and the base pairs are complementary to each other. So in case of RNA, every nucleotide residue has an additional OH group present at the second position in the ribose. Also the uracil is found at the place of thymine. So in RNA, what is the difference is only that uracil is replaced by thymine at the 5th methyl uracil. So this is how the structure of the DNA is. So these are the two strands wherein there is the these are the nucleotides. Okay, these are the nucleotides and these are the pentose sugar which are bond, and the nucleotide has hydrogen bond between them. So which then further forms the structure of DNA. That is how the complementary base pair, that is adenine is complementary to thymine and guanine is complementary to cytosine. So they will be the base pairs has to be complementary to each other, and then there is hydrogen bond which holds the two strands of the DNA. So this is how the structure of the DNA. So how the DNA was discovered? So the Frederick in 1869 first identified DNA as an acidic substance present in the nucleus and named it as nuclein. Then James Watson and Francis Crick proposed a very simple double helix model of the DNA structure in 1953 based on X-ray diffraction data. And then Erwin Chargaff proposed that for a double-strand DNA, the ratios between the adenine and thymine and guanine and cytosine are constant and equal. So this is how the discovery of DNA was done by the scientists. So now we will see some salient features or some characteristic features of double helix DNA structure. So the DNA is a long polymer of deoxyribose nucleotides. It is made up of two polynucleotide chains where the backbone is constituted by sugar phosphate and the base projected inside. So this is how the backbone is sugar phosphate and the base pair is projected inside. The two chains have anti-parallel polarity, that is 5' to 3' and from 3' to 5'. So this is the 5' to 3' end, and the second strand will be having the 3' to 5' end. So it has anti-polarity. So the two chains are coiled in a right-handed fashion and the pitch of the helix is 3.4 nanometer and there are roughly 10 base pair in each turn. Due to this, the distance between the base pair is about 0.3 nanometer. So each turn will have 10 base pairs, okay, which are complementary to each other. The plane of one base pair stacks over the other in double helix. This confirms stability to the helical structure in addition to the hydrogen bonds. So this is how the structure of DNA is. This is the salient features of double helix DNA. So this is a double helix DNA wherein these are the base pairs. This is the sugar phosphate backbone and these are the base pairs which are held by hydrogen bonds. So this is the structure of double helix DNA. So in today's video, what we have learned is what is DNA and what is RNA. We have seen how the structure of DNA was discovered and the salient features of DNA. So thank you for watching today's video. Please don't forget to like, comment, share and subscribe to the Bodhi Academy.